Hello, everyone. For the next 10 minutes, I'm going to tell you a little bit about artificial intelligence for neuropathology prediction, Alzheimer's disease and related dementia subtyping, and prognosis in the Clarity project. And this is joint work with my colleagues at USC and also members of the Clarity team and collaborators Dugu Tosun, Tim Homan, and Dirk Keen's team at the University of Washington. So artificial intelligence can be used for precision medicine in Alzheimer's disease and related disorders by taking all of the aggregated biomarker data from an individual. It could be genetic data, omics data, imaging, or even real-time data, and then calculating a large mathematical model to make a diagnosis or a prognosis or a prediction of their treatment response. There's a couple of reasons you may want to do this, either as a physician to help you make the decision, or as a scientist to discover new features or biomarkers that might help you make the decision. And AI methods are commonly used to discover patterns in biobanks of data from patient populations, where machine learning methods can be trained for differential diagnosis or subtyping, such as in the ATNV framework used in dementia research. Now, why should we subtype brain pathology? Now, one reason is that up to 70 to 80% of patients clinically diagnosed with probable Alzheimer's dementia show at least one co-pathology, such as Lewy bodies or TDP-43 inclusions, cerebrovascular disease, or hippocampal sclerosis. So disease subtyping will help us to identify treatment response or subgroups of treatment responders by stratifying drug trials. For example, a patient with Lewy body pathology may not respond to anti-amyloid or tau targeted treatments. Another reason for subtyping brain pathology is to identify risk factors in the genome and environment by better defining the biology. So genome-wide screening could be used to identify genetic targets specific to each subtype. And this was highlighted recently in Nature, where AI that reads brain scans shows promise for finding Alzheimer's genes. The idea is you could run an AI disease classifier on hundreds of thousands of brain images and then run a genome-wide association analysis on its output to find genomic markers associated with those diagnoses. So early evidence that machine learning could be used to subtype brain pathology came from Dr. Dugu Tosun at UCSF, where she developed a multi-label random forest classifier that was able to infer the presence of multiple neuropathologies that were subsequently confirmed post-mortem, just using in vivo clinical and imaging data, amyloid and tau sensitive PET and MRI. And this was 86 to 89% accurate in identifying TDP-43, Lewy bodies, and CAA, several pathologies that you cannot identify using radiologic imaging when a person is alive. More recent work showed the power of deep learning. So Di Wang and Mohammed Harbez and their team created the DeepSPARE approach, which identified Alzheimer's disease, vascular dementia, and Lewy body dementia with a balanced accuracy of 0.84, 0.84, and 0.62 using random forest support vector machines. And the best method, a deep neural network that performed the best, was 3D ResNet 18. Now, their multi-label classifier was able to indicate multiple simultaneous pathologies in 7 possible combinations, but they noticed that there were discrepancies between a person's clinical and neuropathological diagnosis. Moving on to work by our group, Tamogna Chattopadhyay and our colleagues developed and evaluated 2 deep learning methods to classify 6 autopsy-confirmed pathologies: beta amyloid, tau, cerebral amyloid angiopathy, TDP-43, Lewy bodies, and hippocampal sclerosis. And the methods were a multimodal Y-shaped convolutional neural network, which takes numeric data in on the left and images in on the right, and also an AutoML method called AutoGluon that automatically selects the best neural network architecture to do the task. Now, before we say what the results were, these 2 methods, these 2 deep learning methods, the Y-shaped CNN and AutoGluon, had performed the best in predicting 2-year decline in sum of boxes clinical dementia rating based on brain MRI and NAC, ADNI, and OASIS. The error in this prediction was only 1 point on the SOB-CDR scale, which is close to the inter-rater reliability, so that's why they were picked for this task. So deep learning methods include convolutional neural networks, vision transformers, and vision language models. And they all discover patterns associated with an outcome. For example, does this person have Alzheimer's disease or vascular pathology or something else? And they successively filter regions of the image to identify features that are helpful for these diagnostic tasks. So one of the best performing classifiers of Alzheimer's disease by Bin Liu and Xiaogan Yan is 91% accurate in classifying clinical defined AD, again, based on CNNs that have been pre-trained for a sex classification task and fine-tuned for dementia diagnosis. Now, in work by Emma Gleave in our lab looking at voxel-based morphometry, at least 3 of these pathologies are associated with a significant pattern of gray matter atrophy that you'll see indicated here. So this gives us strong confidence that these patterns could be useful in predicting neuropathology. Now, when these methods are trained, there is a balanced accuracy in predicting all of the neuropathologies of over 0.7, so in some cases as high as 0.85 area under the receiver operating characteristic curve, with the best results being shown for amyloid and tau, but also very promising results for TDP-43, for which there isn't currently an in vivo imaging biomarker. Now, what if we integrate more of what we know about the anatomical patterns of atrophy? So we know that atrophy in clinical AD spreads in a characteristic pattern, similar to the trajectory of amyloid progression or tau deposition progression, but there are still anomalous trajectories and other common patterns of atrophy. So in this film, this was created 20 years ago, we animated the sequence of atrophy that is characteristic of people with Alzheimer's disease, beginning in the temporal cortex, moving into the frontal lobe, and sparing the sensory motor cortices. And because this particular pattern of evolution tracks the spread of amyloid and tau pathology, any departure from this pattern may be indicative of some other pathology. So in an effort to link atrophy subtypes to neuropathology, Conor Owens-Walton in our group used subtype and stage inference, which is an algorithm that identified 3 different atrophy progression patterns from over 4,000 people's MRIs, and no neuropathology was needed to learn this subtyping step. And then he examined how these atrophy subtypes relate to neuropathology subtypes in some held out data that for whom post-mortem data was available. Now, he found that an atrophy subtype S1 with early subcortical atrophy was associated with a greater odds of tau deposition, whereas S2 was a pattern with predominantly limbic atrophy, and this prefigured lower odds of tau deposition. And a much broader pattern of early atrophy, most atypical, was associated with a higher odds of TDP-43 pathology and hippocampal sclerosis. So in summary, there have been several successes. AI methods such as deep learning and sustain can help to identify neuropathological subtypes, but also support diagnosis, prognosis, and staging of Alzheimer's disease and related dementias. Hot topics are using AI-derived diagnoses as proxies for genome-wide target discovery. And there's also interest in using AI to interpret neuropathology slides or images. Some challenges and open questions are that there's limited pilot data so far to train deep learning and sustain methods, but neuropathology prediction will improve with more data. And also the methods I reviewed today focused on T1-weighted brain MRI, clinical and genetic data, but will improve if complementary biomarkers are added, such as FLAIR, PET, or biofluids. Ultimately, these systems will be integrated into an AI-based diagnostic or prognostic system to be used in radiology. So thank you so much to NIH, to the National Alzheimer's Coordinating Center, the Clarity Initiative, and our colleagues at the AI for AD project for making these projects possible. Thank you very much.